So, hello everyone, welcome back to our channel Science Beyond Limit, and I am thrilled to have you here for another exciting educational journey. Today, we are diving into a fantastic as well as one of the interesting topic that lies at the very core of life itself, that is chromosome. Okay, in this video, we will exploring the captivating world of chromosome, especially focusing on their structures and composition. We will mainly focus on these two topics. Okay, we will uncover the secrets hidden within these microscopic structures and understand their significance in determining who we are as individual. Okay, don't forget to hit the subscribe button and the notification bell so you will never miss an opportunity and miss our educational content, miss any of the episode of our educational content. Let's dive into the content, but before diving in, let's take a quick recap about what are the topics we gonna learn today till the end of this wonderful video. So let's see. The first topic is what is chromosome, as usual on the screen. Second topic will be origin of chromosome. The third one is what is chromatin. The coming to the fourth topic, brief idea about nucleosomes and histones. And last but not the least, DNA double strand structures and composition. Guys, this is the most important topic on the whole chapters. You can say that about the double strand structure of DNA and their compositions. The most of the questions from these chapters come from this topic only, that DNA double strand. So the better you understand, the better you learn, and the better you perform on your exam. So let's start. So coming to the first topic is that what is this chromosome? I am telling every time that this is the most important topic you need to carry out. And so what is this actually? See, chromosome is a word or thread like structures. They normally remain coiled. Okay, as you can if you can remember, I don't know if you can remember or not, the oldest telephone have the coiled wire connected to the receiver. Okay, so that type of similar structures we found on the chromosome. Okay, so the definition, if you can tell what is this chromosome. See, the chromosomes are thread like structures made up of protein and single molecule of DNA that serve to carry the genomic information from cell to cell. Now what is this means? What is the genomic information means? See, genomic information is nothing but the genetic informations. We know that our characters, the parent generation's characters is passed to the daughter generation's character. In fact, the characters from one generation of an organism passes on to the next generation of that same organism. Okay, so these chromosomes are the main reasons that carry out these genetic characters from one to the next. Okay, see, these chromosomes now are mostly remarkable structures. Okay, they are the reason why we are generally evolving from one generations to the next. Okay, this normally contains our basic DNA structures, the repetition units that we have studied earlier also. So today even we also gonna learn about that. So let's start. Let's go into the second topic. I hope you understand what is this chromosome. If you clearly identified that, so let's move on to the next topic. Where do you found it? See, this is egg-like structures at the center of the cell. This is a typical animal cell, and there is egg-like structure at the end of the cell where this is the yolk. You can compare it. So what is this actually? This is nothing but this yellow structures, this whole structures. Okay, not the inside one. Okay, I'm clearing it once. Wait, wait, just I am marking it. So this whole structures. Okay, this whole structures including the center yolk. Okay, including this one, this is actually nucleus, and this yellow portion we name it as nucleolus. Okay, so behind the nucleolus where there is a yellow portion, see my red marks. Okay, this red portion area, this is normally filled up with a fluid where we can see this type of structures. Okay, see this type of structures. Even in your previous class, you have drawn this type of diagram, or even you have seen it during study of cell. So this is nothing but that chromosome only. Okay, but we did not name it as chromosome. We named it as chromatin. Okay, so this chromatin only, when they get arranged in definite form, then we name those chromatin as chromosome. Okay, so means why they get arranged. See, during cell division, we know that cell divides from one form to another. Okay, so during that division, this chromatin, okay, this are coil like structures, they got arranged and form a four armed structures. That previous one, see, these four armed structures, they normally formed it. Okay, these four arm structures, they normally formed it, and they form this chromosome. Okay, okay, I hope you understand. This is the basic structure of nucleus. Even see, I am showing this in 2D, but this image is actually 3D. This is the actual nucleus. Okay, you can get a clear idea from this. This is the nucleus. Okay, this is the nucleus. This one is nucleolus, and this portion is filled up with a fluid where we found small thread like structures, that is the chromatin. Okay, I hope this is clear to all of you, everyone. So let's move on to the next topic. Okay, now what is this chromatin? So as earlier I have told that chromosome is made up of DNA and proteins, and chromatin normally arranged himself to form a chromosome. So chromatin is basically made up of mixtures of DNA and proteins that form the future chromosomes only. Okay, these chromosomes are mainly found in all the higher level organisms, and some lower level even organisms also. Many of the proteins, the proteins that we found on these chromosomes are named as histones. Okay, we will discuss about these histones and all these structures in upcoming topic. But first, you know what is this chromatin. Okay, these are normally, just remember, these are the mixture of DNA and proteins only. So this chromatin normally we found in 6 different forms. Okay, see, we can see this chromatin under microscope only during cell division. Okay, it is only visible during cell division. Rest of the time, when the cell is not prepared to divide, we cannot see this cell. So what are the forms actually, when we can check? See, when we can't see, we call those chromatin as fine structures. In some cases we can see there is a box like structures. Okay, small boxes. I don't know if you can notice it or not. See, there is small boxes like structures. Okay, like this structures. Okay, these structures are known as finely stippled. Okay, the lacy structures is known as, means there is a lace, you know, na, shoelace or something, we can compare it to a lace. Okay, so these are the lacy structures. Okay, then this type of structures are known as coarse structures, clumped structures, where the chromatins are clumped at some places. So this type of structures are known as clumped structures. And the last one is smudged structures, where there is a mostly condensed, means you can tell it at the last stage, where the chromatin now is ready to convert to chromosome. Okay, so after this merging stage, this chromatin will get converted to chromosome. Okay, so let's move on to the next topic. So what are nucleosomes and histone? Okay, see, a nucleosome is a basic repeating subunit of chromatin packaged inside the cell nucleus. Okay, I hope you understand. See, this, when we take chromatin, about 150 base pairs of DNA sequence. Okay, and a histone proteins. First, you need to see this image. Okay, see, this is a chromosome. Okay, we have uncoiled it. Just imagine we have uncoiled it, and we will get this type of structures. Okay, this is the chromatin. Okay, now we will more unwind this chromatin. If we unwind this chromatin, we will get a simple these structures like this. Okay, this is also, you can refer it as chromatin. in fact chromatin fibers we can refer this. Now we will more zoom in into these chromatin fibers. What these chromatin fibers actually contain? See, these chromatin fibers are actually packed around some disc-like, button-like structures. Okay, yet thread is normally wind around a button-like structures. This thread is actually nothing but DNA, and the button-like structures are nothing but the proteins, or we refer these proteins as histones. I hope this now it's very clear to everyone. Okay, these proteins are normally, these button like structures are normally histones, and the threads are normally DNA. Now if we more zoom in into the thread, now if we zoom in more into the thread, we will get this type of structures, this type of structures where inside there is a double strand of DNA. There is a double strand of DNA. Clear? I hope you understand easily. This double strand of DNA is very important on our topic. Rest of the topic is also even important, but if you remind it stage by stage, now this is a very important image actually. You can just remember it and you can easily carry out any topic related to this chromosome. I hope you will never get confused about nucleosomes, histones, chromatin, what is chromosomes. Every time just remember this image and you will get a clear idea. Okay, so now let's move on to the topic. What are histones actually? Sorry, see, the histones are normally these button like structures. Okay, these button like structures are normally histones. Okay, now see, there is a four combinations. Okay, actually see, this number is, I can number it, number 1, number 2, number 3 and number 4. Surround this there is a coil like structures which we refer it as DNA, or double stranded structures, that is deoxyribonucleic acid. Okay, now these four structures you can just see there is two layers. This is one layer, this is second layer. I can wait, I will clear the image and you can see now, see, there is one layer and this is just down below it there is another layer. So this histone proteins, now this one is 2 2 2 and 2, total 8 structures. This total 8 structures together form the histone proteins, surround which this line of DNA remains. Okay, so these structures is normally known as histones. Okay, now what is nucleosome? See, nucleosome is nothing but just you can tell it as a accumulations of 150 base pairs of DNA sequence, okay, wrapped around a core of histone proteins. It's just an accumulation that total 8 histone proteins. Okay, total 8 proteins, 8 histone proteins. Okay, these are one each histone proteins, these are the each histone proteins, these 8 histone proteins along with 150 base pairs of, sorry, my handwriting is so bad. Along with 150 base pairs of DNA, we call these structures as nucleosome. Okay, clear? I hope it's clear to everyone. Now let's move on to the next topic, that is our double helix model of DNA. We will more focus into the, we will more zoom in into that where, okay, now the nucleosome, the where present within that nucleosome, or the where that surround that button like structures, that is histones. Okay, when we just zoom in into the structures, we will get this double stranded structures. This is known as DNA, deoxyribonucleic acid. Okay, these are normally found in higher level organisms, but previously this just it is a imagination, or you can tell there is a thought that DNA is updated from updated version of RNA, which is ribonucleic acid. Okay, so this double stranded, actually these double stranded structures was first given by Watson and Crick. Okay, Watson and Crick, for which they become most famous. Okay, these double stranded structures, actually the knowledge of these double stranded structures is so important for us. So actually what is this double stranded structures? See, these double stranded structures are nothing but a combinations of pentose sugar, okay, nitrogenous base and phosphate. How these structures are normally arranged, you can see there is some image. Okay, I will make it clear. See, there are some nitrogenous bases, there are some bases found in this DNA. Okay, what are these bases? It's adenine, cytosine, thymine and guanine. Okay, so you can see these colors na, different colors. I hope you can see it. So these are the different nitrogenous bases. See, there is already written adenine, thymine, cytosine and guanine. Okay, so this nitrogenous base normally combines with each other. Okay, adenine always combine with thymine and cytosine always combine with guanine. Okay, now this adenine combines with thymine always in double bond, but the cytosine combines with guanine always in triple bond. Remember this, this is very important. This is very important. You need to know that adenine combines with thymine with a double bond and this cytosine combines with guanine with a triple bond. Now, how they works actually? See, this is a double stranded structures. Now, I will just take small portion of it. Just imagine this is a double stranded structures and I am taking just one portion, one side of these double structures. Okay, if I zoom in into the structures, what we will get? Okay, I am taking these structures, I am taking these structures. Okay, now what we will get? Okay, see, there are two sides, in fact you can tell two chain, where the chain is made up of phosphate actually. With this phosphate there is a connection of pentose sugar. Pentose sugar means there is total 5 sides. Okay, with this pentose sugar there is a connection of nitrogenous base. I will just mark it as N. Okay, with green, nitrogenous base. Now this is a one strand. Okay, this structure is a one strand. Just wait, I will mark it as white. Okay, and this one as yellow. Okay, see, this yellow is a phosphate, yellow one is phosphate, white one is pentose sugar 5-sided structures, and the green one are the nitrogenous bases A, T, G and C. This can be anything, but we will know how this combination works. Okay, this is a one strand. There are several repetitions. Some just imagine these are number one, and here comes a repetition. The chain continues and this will only form there. Okay, this will only form there, similar repetitions. Okay, hope you understand. Now on the other hand, I'm just taking up the image. Okay, on the other hand, there are similar structure. This another strand, this one is phosphate, connected to the phosphate there is a pentose sugar. I just drawing normally. Okay, this is a pentose sugar and there is another nitrogenous base. Okay, and they remain connected either by double bond. If there is a combination of A and T, this will be the double bond, this structures will be the double bond. Or if there is a combination of GC, this will be triple bond. This bonding depend on the nitrogenous base that are present here. Okay, these structures that are present here, these two nitrogenous base, if there is G or C, this will be triple bond. If there is A or T, this will be double bond. Clear? I hope it is clear to everyone. Clear? So this is the structures. These repetitions gets continued, and in this way the DNA normally takes its form. Okay, I hope it's clear. See, two nucleotide chains whose nitrogenous bases are connected by hydrogen bonds. Okay, one more thing to remember. See, these nitrogenous bases have two names actually. These are purines and another is pyrimidines. Okay, what are purines? purines are A and G are considered as purines, and pyrimidines T and C are considered as pyrimidines. Just remember the fact AT, GC, where the previous one are the purines and the next second ones are the pyrimidines. Okay, I hope everything is clear now. Everything is clear now. The last note for you, this how this DNA actually work actually. Okay, see, this DNA actually makes a RNA template. Okay, how you can understand easily, see, these are the double stranded structures. They just make a copy of a single stranded structures. Okay, and these structures normally makes a protein which carry out in our next generation. This structures actually carry out our genetic characters to our next generations, and in this way the DNA works actually. See, DNA, though we know that RNA just evolved from DNA, but to work, DNA can't perform solo. DNA, even during passing of genetic characters, they make a copy of RNA by the process we named it as transcription. Okay, by the process we known as transcription, and this RNA makes proteins and carry out the genetic informations to the next generation. Okay, so this will be the end of the topic today. So I hope everything is clear. If there is any doubt, you can ask me on comment sections. Don't forget to press the subscribe button and press the bell button so that you will never miss our educational content video. So this much for today. Let's meet everyone on the next video. So till then, bye bye.